Welcome back to the Science Expo Help Series, put on by the Anne Arundel County Public Schools Office of Science. Our contact information is on the bottom of this page. Here, we're going to look at Part 4, projects that deal with potentially hazardous biological agents. As always, you can find more information along with the other videos in this help series on our webpage at www.aacps.org. Here, you can find all of the information to help your student plan, run, and enter a project in the Anne Arundel County Public Schools Regional Science and Engineering Expo held each and every spring. Let's take a look at the forms you'll need for these types of projects. Before we look at the forms, let's talk a little bit about what exactly is a hazardous biological agent. Biological agents are ranked from 1 to 4. This is according to their BSL or their biosafety level, with BSL 1 being the low-risk microbes and level 4 being the ones that are higher risk or the most dangerous. Each level of BSL has its own guidelines for the handling, safety, and equipment needed for these types of projects. Only level 1 and 2 projects are allowed in the Regional Science and Engineering Expo. BSL 1 is considered low risk to personnel and the environment. These types of samples are very unlikely to cause disease, and these types of projects can be completed at the school. However, they should not be completed at home. BSL 2 projects, including those that use blood cells or other forms of higher level bacteria, are considered a moderate risk. They do have a risk of spreading, but it is minimal. The access to the laboratory should be restricted and an autoclave should be available. They do have to have supervision of a qualified scientist to oversee the research, and these cannot be completed at school or at home. This involves the growth and culture of bacteria. These types of projects that are BSL2 must be completed in an appropriate laboratory, usually at a research institution or university. Finally, anything that is level 3 or 4 is off limits. So please do not run projects that are level 3 or 4. If you have questions to the level of your project, please contact your school SRC or the regional science office so we can help you out and make sure you're safe in handling all samples. Again, as with all other projects, you'll want to get prior approval prior to the start of experimentation. This is used on Form 1B, and this form is required for all students, including each and every team member. Prior to gaining approval, the student or his group should have the purpose, hypothesis or goal, materials, and procedure available for review from both their parents or guardian, along with the affiliated FAIR SRC. The FAIR SRC, normally the school department chair or their designee, will fill out and sign box 2A. In very rare circumstances, you may need to get box 2B completed. Box 2B is extremely rare. That is only for students who complete their research prior to the start of the school year at a research institution such as a university or business, or they complete their research prior to the start of the project being introduced in the classroom. Finally, is the FAIR SRC approval. This is signed by the regional FAIR director, usually someone in the science office. Please do not sign box 3. Most projects that use hazardous biological agents are going to require a qualified scientist form in addition to prior approval from the school SRC. There's only a few projects that would be considered exempt from prior approval and the qualified scientist form. You'll have to check the rules to see if this is going to be your project, or contact the school SRC or science office if you're not sure. Gaining the approval of your qualified scientist, you may want to have a few things for them to review prior to signing off. You may want to have the procedure, experimentation, and materials list for them to review. Once they feel comfortable with your experiment, then they will sign on the bottom and date before the start of experimentation. Remember, the date of approval has to be prior to the start of experimentation. If the qualified scientist will not be directly observing your experimentation or data collection, then you will have to have a designated supervisor who will be present. And the qualified scientist can train them in the methods or the different safety concerns to be on the lookout. The designated supervisor, again, also has to have a date of approval signed prior to the start of the experimentation. The major form you will need for this type of project is Form 6A, the one for hazardous biological agents. This is going to be used for any project that uses microorganisms, recombinant DNA, fresh or frozen tissues, including possible cell lines or tissue cultures, blood products, bodily fluids, all of which are going to need SRC approval prior to the start of experimentation. Form 6A should be completed with the qualified scientist or designated supervisor collaborating with the student researcher. It is going to be broken up into 4 sections. The first is the project assessment. This is going to look at the types of experimentation done, the possible procedures, and the biosafety level, along with the containment and disposal procedures. Section 2 is going to look at training. What training or specific help did you receive from either the qualified scientist or the direct supervisor so that you could safely handle the project? Finally, what is the experience or training of the supervisor relating to this area of research? The next section, Section 3, needs to be completed by the qualified scientist or the designated supervisor. Here, they will need to confirm that the project is either BSL-1 or BSL-2 and that a sufficient laboratory space is going to be used for the experimentation. They also may need to verify the cell line or the different types of hazardous biological agents that are going to be used. They will also need to sign off and date their signature prior to the start of experimentation. Finally, Section 4 is going to ask for the local or the affiliated FAIR SRC located at the school to sign and date as well, again, prior to the start of experimentation. The other form you may need is Form 6B. 6B is not required for all projects, but is only required if you're using human or animal tissue samples, which could include fresh or frozen tissue, organs or other body parts, blood, bodily fluids, primary cell or tissue samples, or possible human or animal cell lines. If you're using any of these, you'll need to fill out Form 6B in addition to 6A. You will have to state where did the samples come from, how are they being handled, and where or what type of research facility is going to be used to study them. You'll also have to have a qualified scientist or designated supervisor sign and attest that everything is being handled in the proper environment and under the correct guidelines. They'll have to sign and date prior to the start of experimentation. As always, if there's anything about your project that makes you nervous or hesitant, please fill out Form 3, the risk assessment form. Please do so under the supervision of your designated supervisor or the qualified scientist to make sure that your project is safe and secure for both you and any participants. Remember, it needs to be signed by the person supervising the project experimentation and data collection, and the date of review must be prior to the start of experimentation. That concludes Part 4 of our Science Expo Help series, looking at projects that deal with potentially hazardous biological agents. As always, you can go back and watch our other videos depending on the type of project and forms you're going to need for the Anne Arundel County Regional Science and Engineering Expo. You can also visit our website to find these forms and all of our videos, along with helpful dates to help you plan your project. Finally, if you have any questions, comments, or just general feedback, please reach out to the Office of Science. Our contact information is listed at the bottom of this page. Thank you.